summarize Pat's recent studies, which are really interesting, and these then involve moving again from the human back to the mouse, because this is all based on work in the mouse. So as many of you already know, Pat's been interested in environmental exposures and the association with aneuploidy and with recombination, which is correlated with aneuploidy, for many years. And Steph, I'm just going to say next.
Okay.
So I'm just going to say next now. Okay.
Yep.
So what she's really been interested then in is and particularly focusing on the association between meiotic abnormalities and endocrine disrupting compounds next. And this all started actually in 1998. It wasn't published until 2003, next, where we saw chromosome abnormalities in association with damage to the caging materials that the mouse were housed in, next. And it turned out that these cages and the water bottles were comprised of bisphenol A. And then in a series of experiments, Pat was able to demonstrate that if you dosed animals intentionally to various very small levels of BPA, that you could induce next. In addition to changes in recombination, you could induce synaptic abnormalities, and you could also induce changes in rates of aneuploidy next. And so the story with the female mouse then that is that our lab is still carrying on. And anytime I should say, anytime I say we, it's not really we, it's Pat's group and Pat that has spearheaded these studies. But in any case, what we have seen then is are changes in recombination. So in association with exposure to BPA in the female, surprisingly, we see these increases in recombination rates. We also see changes in chromatin morphology, and we also see changes in the rate of aneuploidy. And this was especially pronounced in the very first exposure where I was involved in these studies, where we saw aneuploidy rates in female mice in controls go from under 1% up to about 10%. So it was really an extraordinary increase in aneuploidy. So it has turned out to be it's turned out really to be a fascinating, and next again, fascinating story, next, and next, and next. Because I'm sorry, back up one step, I'm sorry. So what's happened as a result of all of this, and some of you already know this, is that consumers became concerned about having BPA in baby bottles or in sports bottles, other products, and so really generated by consumers, there's been a concerted march to produce BPA-free materials, and sadly, it in some senses turned out to be a win-win for manufacturers, because you can produce replacement bisphenols that are chemically extremely similar to BPA, and as you'll see a little bit later, have the same kinds of meiotic effects as BPA, but you can charge more for it because you can market this as BPA-free. So it's been an interesting, I think, a societal story to watch. So all of this stuff, the initial stuff was done, Pat did this in the female mouse and is continuing to work on the female mouse. But for the rest of the time, I'm just going to talk about an interesting story that involves looking at the male next, in which we see interesting comparable effects. Next. And so just to remind you very quickly that, that in contrast to the mammalian female, spermatogenesis involves mitotic proliferation both prenatally and perinatally, and the actual guts of meiosis don't occur until puberty in mammalian males. Next. But importantly, the spermatogonial stem cell population is set up in the perinatal or very early neonatal period. In the mouse, this is, I think, around days 1 to 3 after birth. So it's very early that the stem cell niche is expanded and is established. And one of the things that Pat early on was interested in doing next was in asking whether or not, if you exposed perinatal male mice to estrogen, whether or not you might be able to elicit an effect on meiosis. Next. And what she saw was subtle but highly significant decreases in recombination in BPA, in estrogen exposed animals. And we also then saw increases in meiotic errors. Next. And in particular, what we saw was an increase in bivalents that didn't have an exchange between them. And so you could visualize this at diakinesis in chromosomes that were not lined with one another, that had already fallen apart. And in contrast to the female, in which some of these errors can slide through meiosis and yield aneuploid gametes. In the male, next, what happens is you see a decrease in sperm counts. And so all of those things she saw in association with exposure to estrogens. Next. And now, Stephanie, just step through this, and there's going to be a number of different things that are going to come up. And once you keep going, and I'll tell you when to stop. And so what she was interested in was really, I thought it was really clever, but sort of a simple sort of a question, was she was interested in asking questions about not just having a single exposure, but let's just focus on the right-hand part of the slide right now. We might come back to the left-hand slide, but because she was interested in identifying what are called transgenerational effects. If you had an estrogen exposure in one generation, could you see it, a couple of generations later in the grandchild where there shouldn't have been an exposure, at least in males. But what she was more interested in, and that's the thing I want you to focus on, is the right-hand side of this slide, and that is successive exposures over multiple generations. And here's the deal. Most people that look at exposure paradigms in rodent models will expose one generation and then ask what effect it has on the exposed animals or on their offspring, or even another generation down the line. But that's not really how we imagine we're exposed to environmental agents that are now becoming ubiquitous in the environment. Things like BPA, you imagine you're going to have exposures over many generations. And so her question was, if you exposed one generation, and then you exposed the next generation, then you exposed the third generation, would you have evidence for an increase in defects in those lower generations? Might the effects be cumulative over time? Next. And so, in other words, might the third generation be sensitized? Yeah, sorry.
So no, it's my fault. Okay.
We'll take a brief pause, and this is where the excitement is built up. So we have the question: what is the answer?
Okay.
What is it, Jerry?
Is this it?
And that's it.
Okay.
Okay, so the first thing she looked at was really kind of interesting, and that is what we're referring to here as recombination failure. And that means what proportion of cells contained at least one bivalent that apparently, using MLH1 as a marker of crossovers, what proportion of these cells had at least one chromosome pair that was not joined by a crossover? And what you can see here is this, highly significant increase in these kinds of cells, so that when we got to in the third generation of exposure, we actually saw over 30% of cells that had at least one chromosome pair that wasn't joined by a crossover. And these cells are destined to die. We know that from previous studies. So a really marked effect and an increasing effect with successive generations of exposure. So that was the meiotic phenotype. Next. But intriguingly, and this is actually something that was just, it blew us all away when we started seeing this stuff. When she looked at reproductive tracts, when the grad student who was doing this work, Tegan Horan, looked at other reproductive structures, we also saw remarkable increases in abnormalities. And so this just shows vas deferens morphology, and Tegan categorized the vas deferens as either being straight or having these kinked or collapsed abnormal phenotypes. And what she saw next was that in the first generation, we did see occasional kinked morphologies, but after 2 and after 3 exposures, we saw this really abnormal kind of morphology that she called collapsed. And so again, there appeared to be this increase in these kinds of abnormalities, at least in the third generation, over what we saw in the founder generation. And next. And then additionally, she saw a phenotypic abnormality in the third generation, in which I should stop here just for a second and say she made it was really a complicated but intriguing paradigm, so that she had as controls animals that hadn't had any exposures, and then she had different categories of successive generations of exposure. One in which only the founder generation was exposed, one in which the first 2 generations were exposed, one in which the first and the third generation were exposed, and then one in which all 3 generations were exposed. And in those last 2 categories, those are the only kinds of animals in which we saw what she called fibrotic testes. And these were testes that were just, they just didn't even look like testes. So they were extraordinarily abnormally formed. And so she saw that in these kinds of abnormalities then in the third generation animals that had either 2 or 3 generations of exposure. Okay, next. So what does that tell us next? Well, it tells us that we have, apparently as a result of these successive generations of exposures, increased likelihood of meiotic abnormalities, and additionally this really striking and weird increase in specific abnormalities of the reproductive tract. Next. Okay, so is there, so it appears that there might be increased estrogen sensitivity. The other thing that has happened more recently, and I should also, let's go to the next slide. And so I'm not going to go into this, but it does look like the effects certainly on meiosis are associated with the spermatogonial stem cell population and abnormalities there. And we know that from a number of studies that Pat's done. And we know that next, that estrogen exposure and contamination to endocrine disrupting compounds increases the likelihood of things going wrong to this stem cell population. Okay, let's go to the next slide. Remember, 20 years ago, the initial studies that we did involved these cages that had remarkably broken down over a short period of time, and it was due to an accident in the animal facility. Okay, fast forward 20 years. Next slide. And what we're using now is a polymer. We're still using plastic polymers to house animals, but these are caging materials that are much more stable, and they're made out of polysulfone. And next. And annoyingly, now I think it's about 2 years ago, we started noticing abnormalities in these polysulfone cages. And so we saw increasing crazing, and the cages themselves became much more opaque, and you can see the cages in the two bottom cages here appear to be somewhat damaged. And about the same time, next, and I'm sorry, okay, next, we started seeing this, and what Pat was concerned because we had some increases in abnormality rates in our control populations. And so we did a couple things. The first thing was to find out what if anything, might be leaching out of these polysulfone cages. And polysulfone is comprised of these dimers of diphenylsulfone and bisphenol A, and that's what it's made out of. But the chemist who actually then did the mass spec studies to ask what was it that was leaching out of these cages came up with a thing called bisphenol S. So a chemical that's similar to bisphenol A, but not the same thing. And the manufacturer said, that doesn't make any sense, can't have happened because there's not even any bisphenol S in these cages. Well, it turned out that that was based on the idea that you would break if as things degraded, that you would break molecules down into their constituent molecules, where you see these orange marks, X's, and then you would liberate diphenyl sulfone. But in fact, let's go on a couple more. If you change the place where you break the bonds, you can then generate next bisphenol S. And this is apparently what's going on, is that we're liberating, as these things break down, we're liberating bisphenol S. Okay, so who cares? Why are we concerned about this? Well, let's go back to our meiotic assay of crossovers. And one more, Steph, and one more. Yeah, that's it, perfect. So these two bars show control level rates of crossing over, and crossing over levels in male mice that have been exposed to estrogen or to bisphenol A. And these are really relatively tight boundaries, so that animals typically that are control are someplace around 26, and the exposed are at 25. This seems like a very, very small difference, but trust me, we've done these studies for a zillion years, and there really are tight tight error bars around both of these levels. So in any case, these were the historical levels for controls and exposed. And what we started to see at the time that Pat noticed the crazing in the cages the mice were housed in, was that we had a lot of male mice. Next slide. A lot of, is it there? I can't quite see on the left-hand side. Yeah, it's there. Okay. You can see there are a lot of control animals that are down at the exposure level. This is really not what we've seen over years. We don't see this from controls. So what she did was then to replace next was to put in new cages. So we got new cages, and it looked, maybe a little bit better, but there was still a lot of animals. These are non-exposed male mice. A lot of animals that are exhibiting levels that are somewhat surprising in any case. So finally, and this is a process that took a long period of time to sort out. We finally then got new cages and other new cages, and these are still polysulfone cages, but they're not damaged. And we got new mice from Jack's. And finally, once we had done this, these control levels in the non-exposed animals returned to what looked relatively normal. And we've now gone on.
Can I hit next? Can I hit next?
Oh yeah, hit next.
Yeah, there we go.
Yeah, there you go. So so there really has been a shift over time in these different conditions. Okay. Finally, next. So the last thing then that Pat's done more recently and that has attracted a lot of attention is to say, hey, we know there are a lot of replacement bisphenols out there now, including BPS, but we've also got BPF, BPAF, BPX, BPG, BPZ, and Y, I think. So in any case, there are a zillion new replacement phenols that can be put on products, and then you can label them BPA free. So in this recent set of studies, Pat was interested in asking how do these compare to placebo animals that have been exposed to these replacement compounds. What do they look like? And you can see the results there that certainly the replacements appear to be no better, at least with this meiotic phenotype, than is BPA. And in the case of things like diphenyl sulfone, they're actually worse, and significantly worse than is BPA. So this has now attracted a considerable amount of attention because of the fact that this indicates that this new generation of replacement bisphenols may turn out to have as many reproductive consequences as the stuff we've just got rid of. And so with that, I'll just tell Stephanie to go next. And on Pat's behalf, I'll thank all the other people in her laboratory who have done this work. And that's it.
Okay. Thank you. That's great.
Can I ask Terry a question?
Yeah, sure.
I don't know how you can hear me. Hey Terry, it's Bryn. There we go. I just had a question for the multi-generational study that were exposed to estrogen. Can you give more details on what did that exposure mean? Was that like a single dose, or was that like over a long period of time? And was it the same for every generation, kind of like at the same?
Yeah, no, it's absolutely the same. And these are they're and I can't remember the dose, but they're given doses over different windows. And there's one, I think these were given doses from 0 to 12 days. There are different windows where you can elicit this, an effect, and it looks as if, in other studies that Tegan Horan did, she narrowed that window to, I think, from 1 to 5 days postpartum, and I can't remember exactly what the dosing strategy was in these experiments, but it was exactly the same over all generations, and these are extremely low doses. And so I'll just add one thing on really quickly, that the FDA came out last week with studies that involved a lot of different laboratories, both governmental and academic laboratories, and suggested that BPA was unlikely to elicit meiotic phenotypes or any other phenotypes at very low doses. And it turns out that this probably is unlikely to be the case because they didn't go low enough. These are hormones that can act at very low concentration. So it's a huge controversy going on in this field right now as to whether the FDA is in effect covering up work from academic laboratories that have shown clear effects of BPA. Bottom line, the dosing strategy was the same over all generations.
Okay.
Terry, it's Karen. I was curious. Hi. What kind of material does the Jax lab use for their cages, and is that what we should all be using?
Yeah, no, this is the thing. I mean, listen, Karen, you're too young to remember this. But there was a famous movie one time where a guy said, "I have one word for you, plastic." So right now I have one word for you, glass. So the bottom line is, I think they use polysulfone. That's my understanding, and it is much more stable than BPA. And we don't know why these cages, we don't know what happened to cause the cages to do this, but because we'd had them in the facility for a long period of time, and there was no obvious physical evidence of damage.
Dishwasher or hand wash?
Did you
I'm
hear?
sorry? Yeah, and that's one of the possibilities. The first time around, it was a dishwashing detergent.
Yeah.
It was a harsh floor detergent that got used on the cages. And this time around, there's some suspects, but we're not really sure what happened.